We're now going to have a look at the environmental pressures which lead to variation within a population and the changes that can result. We're going to now look at natural selection. So the action of selection on a population in the wild is termed as being natural selection. So we can define natural selection as a process by which heritable traits, whether morphological, physiological, or behavioral, evolve and persist within a population. So natural selection is often referred to as survival of the fittest. So having an increased chance to survive long enough to pass your genes on to the next generation. So different traits may have an advantage in different environments and a disadvantage in others. So it's really important to keep that in mind. Natural selection is about maintaining traits that are advantageous in order to survive within an environment. So there's a little diagram just on the right here, which says, can you see the butterfly? So this butterfly has been able to camouflage onto this tree, and as a result, that's increased its ability to survive. So the butterflies which contain the genes which code for phenotypes which allow them to camouflage would be defined as the fittest in this particular case. So a classic example of natural selection is the growth of resistant bacteria. So antibiotics are used to kill bacteria. However, many bacteria have grown resistant over time, and that occurs based on the mutations in bacteria that can result in some individuals having a resistance against a particular strain of antibiotics. They then reproduce, pass these traits on to their offspring, and then you see colonies of bacteria which are resistant to antibiotics. So we can see a diagram here which shows population of bacteria that are resistant. So capital R stands for resistant. They are the ones that are going to survive when antibiotics are applied. And then the resultant population, or the new population, are all resistant to a particular type of antibiotics. So in this case, the selective pressure of the antibiotics pushes natural selection. So basically this agent for change, the antibiotic, is causing natural selection to happen fast because it's killing off all of the bacteria that don't have that resistant gene. And the advantage of the resistance is then passed on to the next generation so that all bacteria become resistant to antibiotics. Selection pressures. So a selection pressure, and we need to know how to define this, is any factor that influences survival of an individual, a segment of a population, or the species as a whole. So selective pressures only become sort of relevant when there is variation in a population, when some of the population are fitter than others. So selective or selection pressures can act on two main factors: intra-species competition, okay, so competition within a population for mates or the like, or the ability to sort of hunt and find the food, and it can also act on interspecies competition, so animals of different species competing for food, nesting sites, etc. Natural selection is brought about by a range of selecting agents or selective pressures that act on a population. So some examples include physical agents, so things such as climate change or food shortages, biological agents such as infectious disease, predation or competition, or chemical agents such as pollutants, soil and water. There's a nice little table here which shows you variance within a population, an agent of natural selection or a selective pressure that's being tested out, and the outcome of natural selection. Please read through those. So selective pressures and fitness. So selecting agents greatly determine what phenotype is classed as fit versus what is not. So a classic human example is malaria regions and sickle cell anemia. So sickle cell anemia causes issues within red blood cells. It shows incomplete dominance where a heterozygous individual has some sickle cells. So normal cells are susceptible to malaria, resistance to malaria, but may have sickle cell disease occasionally, and people with sickle cells are actually resistant to malaria, but they've got a fatal disease which can cause other complications in their body. So in this particular case, when we're talking about malaria, the person that's got sickle cells is actually going to be fitter than the person who has got normal shaped red blood cells. There's some more information here about malaria and sickle cell anemia. So to sum up this process of natural selection, it comes about due to the fact that there are mutations in a population. So mutation is something that's important. It leads to variation. Without variation, we wouldn't be able to adapt to selective agents or environmental pressures, and therefore we wouldn't be able to survive and evolve over time. So if you're ever asked to sum up these steps involved in natural selection, or why natural selection is important, you need to be able to mention that mutations lead to variation within a population, and that variation allows us to be able to cope with or respond to environmental pressures, where a variant genotype or phenotype has an advantage, and then they are able to survive, pass on their advantageous traits, and evolve. There's a few different types of natural selection. So natural selection can be complete, where any organism with an unfavorable phenotype can't pass their traits on, or it can be partial. So partial is when unfavorable phenotype have fewer matings compared to other fitter phenotypes, but they still exist and they're still able to reproduce and pass on in some cases. Another important thing to note in this topic is gene flow. So changes to a gene pool come about, the combination of genes within a population that is, due to the movement of individuals between populations. So emigration is when organisms move out, which leads to a loss of particular alleles. Immigration is when we've got organisms moving into a population and in doing so introducing new alleles. This may be beneficial or deleterious. So if we think about immigration of thalassemia allele into Australia as a result of sort of European colonization, that's something that wasn't really favourable, but we've been able to overcome it because of the fact that we've got a lot of people who don't have that particular allele within our gene pool. It's just a diagram here showing the difference between emigration and immigration and how you can introduce new genotypes into a population. We also need to be able to define the term genetic drift. So changes in alleles due to random chance events in allele frequencies causing the phenotype of the species to develop in a particular direction. So this is sort of when we're introducing more of one type of allele and less of another. Okay, so this can be impacted by things such as natural disasters, where a lot of a species are lost, and the loss of an allele through chance events of meiosis or founder effect. So when a new population is established, when it becomes separated from another larger group. As time progresses, it is possible that one allele becomes fixed. So in some particular populations, the frequency of allele may become 100% or it may be lost completely. So due to genetic drift, we can see the frequency of alleles can drastically change. So there's a nice little graph that shows this on the left-hand side. So we can see the orange allele decreasing and the blue allele increasing. The smaller the size of a population, the greater the potential impact of genetic drift. We need to look at a couple of different types or a couple of different theories or ways that this can occur. So the first thing that we're going to look at is founder effect. So this is a type of genetic drift that occurs when a small section of a population leaves a larger section and colonizes in another area. So the small group is not representative of the larger population. Okay, so it's only a small section which is being removed. This is a founder group is established, and then the new population that they've colonized in another area, it only contains blue and yellow alleles, as opposed to the original population, which contained blue, green, red, and yellow. The smaller population will become exposed to many different selective pressures, and over time they may evolve into a different species compared to the original population. So founder effect is when we've got a small section of a population being removed, colonized elsewhere. The gene pool that they bring with them is not representative of the whole original population. It's just a small section, and often doesn't have all the alleles of the original population. Population bottlenecks are also another type of genetic drift, and they occur when a non-selective event vastly reduces the number of a species. So a non-selective event is something like a bushfire. It's something that just happens in nature. It doesn't select for a particular phenotype. It literally rips through. Another example would be a volcanic eruption or a tidal wave. Those types of things no organism can survive, so we refer to them being non-selective events. The allele frequencies of the surviving population are not reflective of the original population. The population number may increase in the future, but its genetic diversity will be decreased compared to the original because it was just a small number of survivors. So this tends to have an adverse effect on the population because it can usually just lead to a lack of variation, and even interbreeding can occur because we've just got fewer individuals left in that particular population or that species of animal. It's a diagram here which just shows population bottleneck. So basically we've got a parent population, we've got a bottleneck, so something that's drastically reduced the population number. We've got few surviving individuals. The next generation will build up, but their genetic diversity will be different to the original population. So you can see here already, you can see visually that there's lots more blue than yellow in this particular population compared to the original, which looked much more even. Okay, so we're just going to put together the last presentation and this one and just show how our different agents can lead to variation within a population. So when we have mutation, it leads to increased variation, as do chromosomal abnormalities. Natural selection usually leads to decreased diversity because not all phenotypes will survive. Gene flow, so when we have movement between a population, that increases variation. Genetic drift, founder effect, and bottleneck all decrease the variation or the diversity within a population. We're now going to have a look at speciation, so how the things that we've spoken about can lead to the formation of new species. Speciation is defined as the formation of a new species. So animals are classed as different species when they can no longer inbreed or interbreed and produce fertile offspring with the population they originally came from. So for example, a horse and a donkey can mate and they produce what's called a mule. The mule is infertile. It can't reproduce. It's a different species, and this comes about because of the fact that it actually has an uneven chromosome an uneven chromosome number. So I'm just making this up off the top of my head, but say a horse has 14 and a donkey has 17 pairs, or 17, let's just make that an even number. So it has 18. Okay, the mule would have 7 from the horse and 9 from the donkey, which would lead to 16. And those combinations might be different and they can't pair properly because you've got 7 and 9. So you'd have like 7 lining up on one side and 9 lining up on the other side. You're never going to get every single chromosome that you need to produce a mule. Okay, so separating species. So in order for speciation to occur, organisms must have some level of isolation to prevent interbreeding. So there may be pre-mating isolation mechanisms, so barriers to finding and securing a mate, or there might be post-mating, so barriers that operate after the transfer of a sperm from male to female. So pre-mating isolation. So there's isolation in time, so you're awake at different times in space, live in different locations, isolation by behavior, so they don't recognize each other's courtship rituals, isolation by anatomy, so different parts. There might also be post-mating isolation, so there might be incompatibility of gametes that happen, and that causes species not to be able to develop. There might be zygote mortality, so fertilization occurs, but the zygote doesn't develop. There might be inability of zygotes to develop an embryo, but no further than that. And some hybrids are just sterile or infertile, like the mule, because the chromosomes can't pair, so you can't produce members of that species. Speciation is a vital component of evolution. If we didn't have the formation of new species, our populations would not be able to continue to evolve and we wouldn't be able to survive on Earth. We need to be able to recite the steps in speciation. So I've written them down here for you. So in order for speciation to occur, the first thing that we need to note is that variation exists within a population. By the way, this is a common exam question. When a population of a species splits, there may be different environmental pressures or environmental differences which lead to different selective pressures. As these changes progress, random chance makes it highly likely that a significant development in phenotypes will occur in the two different groups. So there'll be phenotypic change. And eventually the two populations can no longer breed and produce fertile offspring. So with these questions in an exam, you need to definitely mention variation. You need to definitely mention different selective pressures or different environmental pressures, which lead to different phenotypes being produced. And the final thing that you need to mention is over time or eventually, this new population can no longer breed and produce fertile offspring with the previous population. There's a couple of different types of speciation. So there's allopatric speciation, and this occurs when a population which initially has variation is split by a physical barrier. So allopatric speciation is speciation that occurs due to geographical isolation. I usually like to think of this one as like an earthquake happens and a valley forms between two parts of a forest, and as a result, there's two new populations. Over time, different selective pressures, and those plants or animals on those two sides evolved to be different species. This doesn't happen overnight, it happens over hundreds of years. Sympatric speciation. This happens when there's no geographical isolation. The population's not split, but they just diverge out anyway.